Hi everyone, my name is Chaoran Yang. I'm from Victorian Heart Institute, Monash University. Today I'm honored to give a presentation on how to use fecal metaproteomics to study the association between the cardiovascular risk and the protein expression profile of human gut. First of all, why do cardiovascular diseases matter? According to the Global Burden of Disease study, currently cardiovascular diseases are the first burden of diseases worldwide. According to a recent study, currently we are losing more than $400 billion every year due to cardiovascular diseases, and this amount is forecasted to be tripled by the end of 2050s. Most importantly, currently 1 out of 4 people are dying due to cardiovascular diseases. The development of cardiovascular diseases is very complex, and it is most tightly connected to the environmental factor like diet intake. Previous study has demonstrated that there exists a very complex interaction network between the environment, the gene and protein expression profile of human, and also the gut microbes, which means the microorganisms living in human gut plays a very important role in the development of cardiovascular diseases. Based on previous findings in this study, we would like to study the interaction between the human and gut microbes for the development of cardiovascular diseases in the gut. Compared to traditional technique like metagenomics, the metaproteomics to capture both human and microbe proteins from the human gut, and therefore it is especially suitable to study our question. And here we have recruited 63 participants from the Victoria state of Australia. Besides, we also recruited 26 patients of HFpEF. HFpEF is a specific type of heart failure. It is tightly connected to high blood pressure, and also it is more prevalent in female, but the development mechanism of HFpEF is still largely unknown. Here we collected the stool samples from these participants, and we performed metaproteomic analysis. By applying unsupervised machine learning method based on the human protein expression profile, we have found two distinct clusters, which also have a quite different protein expression profile for the gut microbes. Next, we analyzed the cardiovascular profiles of these two clusters, and we found that one cluster has 9 unit higher nighttime systolic blood pressure in nighttime, and which is significant. The nighttime systolic blood pressure has been considered as a better choice compared to office blood pressure because it could avoid problems like office hypertension. Therefore, we named the cluster with higher nighttime systolic blood pressure as the high-risk group and another group as low-risk group. We also found that the high-risk group has 6 difference of the low fiber and also the potassium intake compared to the low-risk group. Next, we have analyzed the differentially expressed human proteins between the low-risk group and high-risk group. For the underrepresented human proteins in the low-risk group, we found a term related to the conversion of angiotensin-2. Angiotensin-2 plays a crucial role in the raise of blood pressure, and it could be converted by 2 enzymes, CTSD and CTSG, which are underrepresented in the low-risk group. Besides, we also identified the terms related to inflammations significantly enriched in the underrepresented human proteins, which suggests that the inflammation is suppressed in the gut of low-risk group. suggesting these two mechanisms may related to cardiovascular protective function of the low-risk group. By contrast, for the overrepresented human pollutants, we have found that the terms related to metabolisms and also the pancreatic secretions has been significantly enriched, suggesting a better metabolic status of the low-risk group. Subsequently, we analyzed the differentially expressed microproteins between the low-risk group and high-risk group. We found that an enzyme called phosphate acetyltransferase is significantly upregulated in the low-risk group. This enzyme is key for the synthesis of short-chain fatty acids, suggesting an increased production of the short-chain fatty acid in the gut of low-risk group. The short-chain fatty acid is the main beneficial product of dietary fiber fermentation by gut microbes, and short-chain fatty acid could lower blood pressure. Finally, we have constructed a co-expression network between the human and microproteins, and we found a network which is significantly downregulated in the low-risk group, which contain both microproteins and also the human proteins, widely known for their cardiovascular risk, like CTSD and PRG2, suggesting this downregulation of this co-expression network plays a crucial role in the cardiovascular protective effect of the low-risk group. Collectively, all this data demonstrated that the metaproteomics is a new choice to determine the gut health in humans. Next, we have constructed a machine learning model based on the healthy cohort, and this plot here demonstrated that this model is well trained because the area under curve is 0.9. Next, we used this model to predict high risk and low risk group in the HIF cohort we collected. And we found that the high risk patients forecasted has a significantly limited ability of exercise, which has been considered as the gold standard in clinic to predict worse clinic outcome in long-time term. To demonstrate that our model could also be applied to a larger population to predict the blood pressure and also long-term cardiovascular risk, we have calculated circulating protein scores based on the metaproteomics analysis of the human proteins, and we weight this score by the weights generated by the machine learning model and applied this model to UK Biobank, which is a large population study. We found that for those UK Biobank participants has higher circulating protein scores, which means they have a similar protein expression profile compared to the low-risk group we have identified. They have a significantly lower systolic blood pressure, and also they have lower cardiovascular risk in long term as suggested by this Cox hazard ratio model. So in conclusion, here by using metaproteomics and applying a machine learning model, we have successfully identified two groups with distinctive cardiovascular risk associated with their protein expression profile in gut. And this suggests some relevant development mechanisms of cardiovascular diseases associated with human gut. Thank you very much for listening. Here, I would like to thank all my collaborators and also all members of the Hypertension Research Laboratory. Thank you very much for listening, and please let me know if you have any questions. Thank you.